So this is the work that I'm now presenting is the work that I did during my PhD in London in the lab of Guillaume Charras. And we heard about indentations on cartilages, bones. Here I'm going to more focus on single cell. Just I will have a couple of slides for motivations. Why do we need to know mechanical properties of the cell or cell mechanics? This is a dividing cell. The green, the reds are microtubules assembled in the middle of the cell, get connected to DNA or chromosomes through these dots, which are called kinetochores. There are two green dots at the end also. They call microtubule assembly. They attach to these chromosomes, start oscillating the chromosomes, and they induce forces and detach the two chromosomes and make the cell to divide. So this is the basic function of the life. The important question is, how does genes control mechanics of cell division? And if mechanical properties of the mechanical properties of the cell and the cytoplasm affect the cell division. In a recent study, we found that the mechanical properties of cytoplasm are very important. Patients with low white blood cell counts have cells. The patient with low white blood cell count. This low white blood cell count is because of a lack of or delays in cell divisions, and they develop cancer. We found that this delay in division is because of changes in intercellular properties.
Does anyone actually measure this force of?
Yeah, there's a science paper 2013 or 2012 that shows these oscillations are not random; they are very precise, and they work on a very precise time scale, and they are oscillating in a very precise manner. And if there's any perturbation to this oscillation, the division is gonna fail. Just as another example, going to a bit of larger scale, extracellular, to the extracellular scale. Your heart valve cells beat over your heart beat over 3 billion times over your life. The heart valve cells experience 10 to 30% strain with the strain rates of 400 to 1,200. So how those heart valve cells can stand such extreme condition? The answer to some of these questions is coming from cells are made of what material. So the first question we need to ask is, what is the mechanical properties of the cell? Since, I don't know, 10 years, 20 years, which this topic of cell mechanics started, everybody now knows that cells are not elastic, they are not viscous, they are viscoelastic. And I need to mention here, the viscoelasticity is time scale dependent. Every material is viscoelastic. Everything will flow over time. When we are talking about viscoelasticity of the cells, we are talking about few hundreds of milliseconds to few hours to days. We are not talking about long time scales viscoelasticity. Another concept in measuring mechanical properties of the cell is the length scale. The mechanical properties that we are looking can be varying a lot depending on the tool that you are using to measure the properties. So if you go to intercellular scale, in the scale of proteins and single molecules, you are talking about nanometer scales. If you go to higher scale, in the tissue scale, you are talking about millimeter scale, and the cell itself is in between these two length scales. So there are two important parameters that we need to consider in characterization of cell mechanical properties: length scale and time scale. We cannot say cell is elastic, viscoelastic, poroelastic, whatever. So unless we determine on what time scale, what length scale we are talking about. People have developed different models that are like at least 20, 30 models to, let's say, fit the experimental data or explain the measured mechanical properties of the cell. Some of the models are like top-down models. For example, you can fit any experimental data to a spring dashpot model, or recently it is suggested that the mechanical response of the cell follow a power law rheology. And there are other models like bottom-up models. So you want to make a models to create or to describe the response of the cell from its constituents, from its microstructure. There are lots of problems with both models. This is too much complicated because the cell is not simply made of simple polymers. It's made of polymers plus macromolecules plus lots of stuff inside it. The problem with these models is you can fit any kind of material with this kind of model, including iron, I don't know, or aluminum, or cell, or whatever. So let's forget about these models. Let's just think about physical picture of the cell. Here is a SEM images of a fish cryocyte, migrating fish cryocyte. If you look more closely to the image, if you zoom in, you can see there are some filaments, there are some macromolecules, and there are some holes inside this SEM image. And the water is diffusing in between this structure. So 70% of the water is cytosol, and the rest is this porous, we can call it elastic matrix, consists of filaments, macromolecules, and other stuff inside the cell. So we can think of the cell like a water-filled sponge very simply, just taking into account this very simple physical picture. Considering that physical picture, there has been recent experiments that suggest the mechanical response of the cell is really poroelastic. So if you have this is a HeLa cell, you apply hyperosmotic pressure by applying different osmotic different medium on part of the cell. Let me see if the movie is working. There's the movie. I need to click on here. So that line is a hyperosmotic pressure on part of the cell, and you can look at the stress propagation inside the cell. Looking at this stress propagation suggests that it is very slow compared to pure viscoelasticity, and suggests that the stress propagation is following a poroelastic rule, which is dependent on the length scale when you apply this hyperosmotic pressure. Another problem is that this is in these blebbing cells. These cells naturally bleb, so they make these balloons. So the explanation for the formation of these blebs is there is a creation of hydrostatic pressure at the point of the bleb, and that hydrostatic pressure builds up because of poroelastic nature of the actin cortex near to the membrane, and this buildup of the pressure pushes the membrane. If there's a little bit of instability in the membrane attachment to the cortex, pushes the membrane out and causes these blebs to form. So this is another phenomenon in the cell that suggests poroelastic nature of the cell. When I joined the lab, Guillaume lab, I was asking, okay, these are the experiments that suggest cell is poroelastic. Can we measure cell poroelasticity directly? Like, I don't know, people measure bone poroelasticity, cartilage poroelasticity. Because cell is so soft, it's hard to use conventional techniques. So we need to use the AFM, which the previous speaker talked about it in detail. I'm not going to talk about it, but this is the image of tapping mode image of a HeLa cell. This is the AFM tip. There is a latex bead, 15 micron latex bead, which we glue it on the cantilever. And this is the HeLa cell. This is a cross section. The bead is 15 micron. The green is a cell membrane. We indent the cell with this bead. So here's our experimental setup. We press the cell with high speeds. I need to repeat this. We press quite high speed, and we hold the bead on the cantilever on top of the cell, and we look at the force relaxation. So looking at the pressing mode or indentation mode, we can kind of have an idea of about elastic modulus. Looking at the relaxation mode, this is the most the complicated part. You can fit this relaxation mode to spring dash mode models, or recently it is shown that it's better fit with power law model. But how about poroelasticity? How can we differ? How can we interpret this data to a poroelastic model? For this, I'm going to talk a little bit more about what's important in poroelastic material that is unique to poroelastic material. Poroelastic theory was first developed by Bio in 1940, and then it was reframed by Mao 1980. It's basically 4 constitutive laws. The strain-strain relationship is similar to strain-strain relationship in a solid. The only new term is this pressure. Here, there's a Darcy transport law, so it basically says if you have a flow of fluid inside your material, how would it be resisted by the pores inside the material? There's a continuity equation, there's an equilibrium equation, the total force needs to be equilibrated inside the system, and if you combine these equations, you will come up with a diffusion equation. Let's go to a much simpler one-dimensional model. Suppose that we have a poroelastic material in a confined environment, and we apply a force, a sudden step force on top of the plunger, and the only way for the fluid to get out of the sponge is on top of the plunger. If you write down those complicated equations for this simple model, you will come up with these 4 equations, and if you do a little bit of calculation for sudden step change, you will come up with this pressure diffusion equation, or with this strain diffusion equation. So the important thing in this equation is if you apply sudden force, you will come up with a diffusion equation. So basically, this diffusion constant is how fast your build-up pressure is propagated inside your poroelastic or sponge. And the diffusion coefficient can be related to the hydraulic permeability and the shear modulus of the material. So what's special about poroelastic material? If you have some gauge in this system, and you measure the relaxation of the pore pressure inside the system, you will see that the pore pressure relaxes exponentially. So this is the first thing. The pore pressure relaxes exponentially. And the exponential decay is correlated to a diffusion coefficient, poroelastic diffusion coefficient, and the length scale, the distance between the gauge and the top plunger. So there's a relationship between time scale and length scale. This is very unique for poroelastic material. You cannot find this for simple viscoelastic material. And another important thing is if you rewrite this equation, you will have a relationship between diffusion coefficient and the macrostructure, between the elastic modulus, the viscosity of the interstitial fluid, and the pore size. So it's kind of a coarse-grained bottom-up approach. So if you can measure these parameters, you can have an idea of the pore size inside your system, which is unique.
It's nice to separate the permeability from the elasticity.
Yeah, that's what I'm going to talk a little bit more. So I'm going to show a couple of slides about how we can say cell, how we proved or how we explained cell is behaving like a poroelastic material. So if you do indentation test on a polyacrylate hydrogels, which are a well-characterized poroelastic material, and if you plot them on a log-log scale, you will see that they are having exponential decay. So if you do polyacrylate hydrogel with different indentation depth, you'll see these shapes. If you do this on cells, you will see that the first part is very similar to exponential, but the latter part is continuing. It doesn't go exponential. It continues like a power law. So from this graph, you can see that at short time scales, which are of order of like 100, 200, I don't know, 500 milliseconds, the curves look like more like exponential, and at long time scales, it looks like a power law. The second important and nice characterization of poroelastic material, if you do indentation on polyacrylate hydrogel with different depths, and then if you normalize the force with respect to with a function like this, with respect to the final force, you won't have the collapse of the curve on a single curve. If it was a normal viscoelastic material, all the curves should collapse onto single curve because it doesn't relate it, the response shouldn't be related into indentation depths. If you normalize the curve with respect to indentation depth, then they're gonna collapse into single curve. For cells, we did the same experiments. For example, MDCK, which are kidney cells. We did indent the cell on with different indentation depths, and then we normalize them. We normalize the force with some function like this, and then we normalize the time with the indentation depth, and the three curves collapse onto single curve. So these two first experiments suggest the response of the cell is similar to hydrogel, which are well-characterized poroelastic material. The next step was how to measure poroelastic properties. So we know from force indentation curve we can measure the elastic modulus. How about relaxation phase? There's an analogy between this indentation test and this model. You can run finite element simulation of indentation of poroelastic material, which are done in these groups in Harvard and in Cambridge. Michelle is here. And you can find some master curves. It's basically the same. The force decays exponentially with the time scales there. And the time scale is related to the length scale and the diffusion coefficient. The length scale is very important. The length scale is a contact area. So it's not indentation depth. You can have a large contact indenter, small indentation depth, or you can have small indenter with a large indentation depth and get the same effect. So what we did is we fit the indentation part to a Hertzian model, and we fit the relaxation part to a function which comes from finite elements simulation, and we calculated the diffusion coefficient by doing this. So we wanted to know which part of the cell is contributing to the pore size and poroelasticity. So what we did is we applied different perturbations. For example, we did hyperosmotic or hyposmotic pressure on the cell. So doing a hyperosmotic or hyposmotic pressures make the cell swell or diswell and change the pore size inside the cell. And we measured the elastic modulus and diffusion coefficient after and before treatments, and we compared let's say the diffusion coefficient and the pore size. Or we use different drug treatments to perturb actin, microtubules, keratins, all different types of microstructure inside the cell, myosin motors, and see which one is playing the major role in setting the pore size. Just a quick overview of, for example, swelling, deswelling experiment. So if you swell, let's say, if you apply hyperosmotic, for example, if you apply a hyperosmotic pressure on a cell, the cell shrinks, so the volumetric pore size decreases. If you measure the diffusion coefficient and elastic modulus, they have this kind of behavior. we couldn't exactly have this relationship between diffusion coefficient, this square, we couldn't find a square. But the trend is qualitatively the same. So when you increase the pore size, the diffusion coefficient increases, which means the cell is less permeable or the pores are bigger. And if you shrink the cell, you have smaller pore size, the diffusion coefficient is decreasing. It means it's less permeable and the pressure is propagating slower.
You get an absolute value of pore size?
Yeah.
Rather than
Yeah. So if you do a little bit of calculation, there's a lots of assumption. We calculated the pore size inside the cell, the average pore size, because this is the effect of cell cortex plus inside the cytoplasm. We are doing 1 micron indentation. So we calculated to be 15 nanometer. And we confirmed this by injecting quantum dots of the size of the 15 nanometer inside the cell. When you do inject these particles, when you apply, for example, a little bit of hyperosmotic pressure, this particle stops moving. And when you apply...
Where does the particle stop moving?
Because...
No, where?
Inside the... We don't know.
We don't know where.
It's a whole cell, yeah. So the next step was to perturb the cell with different drugs, and we found that actin is the main determinant of the pore size. So we, for example, use drug like tronquine to depolymerize the actin, or we constitutively overexpress the actin, as seen here. So there's a overexpression of actin network in here, and we found that if you increase actin polymerization, you will have smaller pore size, you will decrease the diffusion coefficient, you will increase the elasticity, and so on. So coming back to the first slide that I was talking about, internal properties of the cytoplasm, we showed that by having more actin inside the cell, you will have the smaller pore size, and it could affect the cell division because you have more viscous environment and more obstacles for the cell to divide. So here are the conclusions of my talk. So we could measure the poroelastic properties of the cell at short time scale. We found that the poroelastic properties or rheological properties are scaled with the scaling law, and we found that F actin is the main determinant of the cell mechanics. If we do a little bit of calculation on that scaling law, we can find this nice scaling relationship. So you can correlate the diffusion, the poroelastic diffusion coefficient to the diffusion of particles inside the cell, to the average pore size, to the bending rigidity of the polymers, to the size of particle and other parameters. So going back to the previous problem, to the hard valve cells, how can hard valve cells withstand such an extreme condition? So the poroelastic nature of the cells can help them to withstand such extreme condition, to act like a shock absorber. And so this is just a motivation how cell poroelasticity could be important in physiology. I would like to thank my advisor, and also we're closely collaborating with Professor Mahadevan in Harvard, and I would like to thank other friends who helped me to do this study and my funders. Thank you.